Hello everyone, this lecture video is all about oxidation of fatty acids, and the learning focus is for you to be familiarized with the four-step beta-oxidation pathway by which the fatty acids are converted to acetyl-CoA and how the cellular locations where the chemical reactions of the beta-oxidation pathway occurs. Remember that these fatty acids are obtained from the triacylglycerol molecule. So first, the fatty acid must be activated by binding to coenzyme A. Second, the fatty acid must be transported in the mitochondrial matrix by a shuttle mechanism. Third, the fatty acid must be repeatedly oxidized, cycling through a series of four reactions to produce acetyl-CoA, the reduced form of flavin adenine dinucleotide, and the reduced form of nicotinamide adenine dinucleotide. So let us start with the fatty acid activation. So this is your free fatty acid from the triacylglycerol molecule, and the outer mitochondrial membrane is the site of fatty acid activation. Remember that. So here the fatty acid is converted to high energy derivative of coenzyme A. The reactants are the fatty acid, coenzyme A, and a molecule of ATP. With the help of the enzyme acetyl-CoA synthetase, it will attach the coenzyme A to the free fatty acid, and you will have the acetyl-CoA. So we will be using 2-phosphate from ATP, and you will have the adenosine monophosphate. So as the usage of energy as the activation, you will be having 2 inorganic phosphate plus the acetyl-CoA. And this is that. The hydrogen will be a hydrogen ion, free hydrogen ion. So you will now have the acetyl-CoA and 2 inorganic phosphates. And then the difference between the designation of acyl-CoA and acetyl-CoA is that this acyl here, refers to the random length of fatty acid carbon chain that is covalently bonded to coenzyme A, whereas the acetyl refers to a 2-carbon chain covalently bonded to coenzyme A. And then the second stage of the oxidation of fatty acid is, of course, the fatty acid transport. Remember that the fatty acid activation occurred in the outer mitochondrial membrane. So the acyl-CoA is too large to pass through the inner mitochondrial membrane to the mitochondrial matrix. This is the intermembrane space, so that is too large. So what will happen here? Why do the fatty acid need to enter, or why do the acyl-CoA need to enter the mitochondrial matrix? Because here in the mitochondrial matrix is where the enzymes needed for fatty acid oxidations are located. So because the acyl-CoA is too large, we will need a shuttle mechanism involving the molecule carnitine. So this carnitine affects the entry of acyl-CoA, meaning to say, the acyl group is transferred to the carnitine molecule. So the acyl-CoA from this acyl-CoA here, right? This is the acyl-CoA. It will attach to the carnitine. So now, carnitine plus the acyl-CoA, you will have now the acyl-carnitine. So this is the acyl group that attached here. And it will enter now to the shuttle, to the inner mitochondrial membrane up to the mitochondrial matrix. This is the mitochondrial matrix where the enzymes needed for the oxidation of fatty acids are located. So what will happen here? Next is, once it entered the mitochondrial matrix, the acyl group is then transferred from the carnitine back to CoA molecule. It will remove the CoA here, and you will have now the acyl-CoA and a carnitine shuttle. So the carnitine serves as just a jeepney transportation for the acyl-CoA as a passenger to enter the mitochondrial matrix. So it's just like a transportation in which it will lead, or it will bring the passenger acyl-CoA inside the mitochondrial matrix for fatty acid oxidation. And then the third stage is the reactions of the beta-oxidation pathway. We can define beta-oxidation pathway as a repetitive series of 4 biochemical reactions that degrade acyl-CoA to acetyl-CoA by removing 2 carbon atoms at a time, with flavin adenine dinucleotide, the reduced form, and the reduced form of nicotinamide adenine dinucleotide is also being produced. So this process is a 4-reaction sequence that generates the acetyl-CoA molecule and an acyl-CoA molecule that has 2 fewer carbon atoms. Let us start. So this is for the saturated fatty acid. The first step is dehydrogenation. This is your acyl-CoA that has been transported in the mitochondrial matrix, and with the help of the oxidized form of flavin adenine dinucleotide and the enzyme acyl-CoA dehydrogenase, it will remove the hydrogens in beta form, meaning to say it will in beta and in trans form. When we say trans form that is in opposite places, right? Opposite places. Unlike in cis form, they are aligned in the same carbon. So here in trans form, the hydrogen is placed in the opposite carbons, in different carbons in opposite directions. So here in acyl-CoA, with the help of this enzyme, it will remove hydrogen in an opposite manner. So it will remove this hydrogen in beta and in the alpha. So you will now have a trans-enoyl-CoA. The difference is that in trans, the hydrogens are placed oppositely. So here you only have a single bond, but once you have removed the hydrogen here, you will have a double bond here in the beta carbon. So how do you know that this is a beta and alpha? So the designation of alpha and beta depends on the location of the carbon in the functional group. Here in this case, we have here a carbonyl functional group. This is the carbonyl. And the carbon next to that, from right to left, is the alpha. And the second carbon is the beta. So you have here now the trans-enoyl-CoA. Okay. That is the designation for the positioning of the carbon in trans. Trans because you have the hydrogen at the opposite and you remove hydrogen at the opposite manner in the alpha and beta. Now this trans-enoyl-CoA will proceed on the step 2 of the beta oxidation pathway, which is the hydration. In this manner, the trans-enoyl-CoA, with the help of the enoyl-CoA hydratase, an enzyme, and addition of water, it will add water in the alpha and beta carbon. So a hydroxyl will be placed at the beta carbon and a hydrogen will be placed at the alpha carbon. So you now have the left-handed beta hydroxyl CoA. You have your hydroxyl and the hydrogen, which is from the water that has been added on the reaction. And now you have the left-handed beta hydroxyl CoA, which will undergo the step 3, in which this is the second dehydrogenation. We will remove a hydrogen here in the second time. So the left-handed beta hydroxyl CoA, with the help of the enzyme beta hydroxyl CoA dehydrogenase rather, which will remove hydrogen, and the coenzymes oxidized form of nicotinamide adenine dinucleotide. When you remove a hydrogen from the bottom of the beta carbon, okay, this carbon here at the hydroxyl group of the beta carbon will be removed, and at the bottom as well of the beta carbon, okay, you will have a double bond here, which is a carbonyl group, 2 carbonyl groups, and then this one and this one, and the CH2 here, we just compress the hydrogen here, 2 hydrogens, that's why you have a CH2 here. So you will now have the beta ketoacyl CoA as your product in step 3, second dehydrogenation. And the hydrogen that has been removed here, okay, will be placed at the oxidized form of the nicotinamide adenine dinucleotide. Therefore, you will be producing a reduced form of nicotinamide adenine dinucleotide and a free hydrogen ion from the hydrogen that has been removed at the hydroxyl top here. This one. And then this beta ketoacyl CoA will proceed on step 4, or the theolysis. From the lysis itself, it means breakage of the bond from the derivative of coenzyme A. So this beta ketoacyl CoA will be splitted the bond here with the help of the enzyme thiolase, which will break the bond. And an addition of coenzyme A sulfhydryl will be added. You will now have acyl-CoA with 2 fewer carbon atoms because we have removed carbons here. There is a breakage. Coenzyme sulfur will attach to this one. So you will now have the acyl-CoA with 2 fewer carbon atoms. And then the hydrogen in this coenzyme sulfhydryl will attach in this part. So you will have the CH3 instead of CH2, and that is the acetyl-CoA. The products of step 4, thiolysis, is 1 acyl-CoA with 2 fewer carbon atoms and 1 acetyl-CoA. Now, the new acyl molecule is recycled through the same set of 4 reactions again. Because if you will remember, it will proceed on the step 1 again. The new acyl molecule, it will undergo the first dehydrogenation again. And then, it will yield the same set of 4 reactions again, which will yield another acetyl-CoA, a 2-carbon shorter new acyl-CoA, and then the reduced form of flavine adenine dinucleotide and reduced nicotinamide adenine dinucleotide. And the recycling will continue again and again, and it will occur again and again in the beta oxidation pathway until the entire fatty acid is converted to acetyl-CoA. So you know that acetyl-CoA is being used in the common metabolic pathway. So that is all for the oxidation of fatty acids.